Thank you guys so much for joining this webinar series. This is the ABIC Community Connect webinar series. It's a monthly webinar series where we invite different speakers within the imaging community to speak on their various areas of expertise, with the aim of providing you with tools, skills, and information that will empower you and your research. If you have not seen the previous episodes of this webinar series, which is episode 1 and 2, they are available on the ABIC YouTube page. So please head over there after this webinar to watch those. And so with no further ado, for this episode, we're going to have a 2-part episode where we talk about how to plan an experiment and how to choose the right microscope for your experiment. Doing the how to plan an experiment is Michael Reiche. He's the microbiologist and imaging scientist here at the University of Cape Town. He manages the Africa Microscopy Initiative, and he is the co-founder and executive committee member of ABIC. And how to choose a microscope for your experiment will be presented by Soné Hendricks. She's the Division Manager of Microscopy at Carl Zeiss, and I think the perfect person to tell us which microscope to use. A couple of housekeeping rules before we get started. As you guys will have noticed, you were muted upon entry. You will be allowed to unmute yourselves during the Q&A sessions if you would like to. Alternatively, you can post your questions in the Zoom chat or in the Q&A box during the webinar, or you can reserve them for Q&A sessions after each presentation. And so this is how the program is going to look. After each presentation, we'll have a 5-minute Q&A session where you'll either be able to unmute or send your questions in the chat box. And so without further ado, Mike.
Hi, everyone. My name is Mike Reiche. As Precious mentioned now, I'm a microscopist and researcher at the University of Cape Town, and more recently, the operations manager of the Africa Microscopy Initiative. And today, as part of this short webinar, I'm going to be talking about how to plan a microscopy experiment. And sorry, I'm getting feedback from someone. Apologies. My talk today is going to be mainly based on advice and tips that I've kind of gathered during my time in performing and helping design experiments, as well as a few insights that I've been exposed to while working with AMI project proposals, as well as just concepts that have come up in discussions with various discussions with community members. Many of you are actually attending this webinar right now. I know that the audience of these talks is very broad and diverse, but a lot of my talk is going to be focusing on biologists that want to do microscopy for the first time, but bear in mind that applications in microscopy are very diverse. So what I talk about might not always be relevant to you, but I'm hoping that you might still be able to gain some insights. I also want to start by being upfront that some experiments can take a few hours to perform, and they don't require that much planning. But other experiments can take years and months to progress from conceptualization to the ultimate realization of that experiment. It ultimately depends on what is available to you and what you're trying to do. But because of this massive time and resource commitment, it's very important to plan your experiment as best as you can. And then kind of just to highlight that there is a lot you can do with microscopy, and I'm going to be relatively general in my talk now, but I'm hoping that you'll be able to identify tips and tricks and ideas that will benefit you in your pursuit of microscopy experiments. So different types of experiments have different needs. And the first thing you need to do when you're planning your experiment is to ask yourself some questions. What are you trying to see? What do you want to see in your sample? It's the most important driving factor of any experiment that you want to design. And then the very next question you should ask, is a microscope the best or the most suitable approach for doing what you want to do? Microscopy captures spatial information and biological context. If there is no need for you to maintain spatial information or biological context, then microscopy is probably not the best tool. And there are other tools that many researchers can resort to. And that includes flow cytometry, mass spectrometry, Western blots, qPCRs, many, many things. There is a significant overlap between what you can do with microscopy and many of these applications, but sometimes it's easier to not do microscopy and to do an existing pipeline performing a northern blot, for example. So the next question is, what is the goal of your experiment? Are you trying to establish whether your sample works? Is your staining protocol working? What does your sample look like for the very first time? Or are you trying to follow a more directed experiment where you want to identify data that you can use for insight? Because they have different ways at which you would approach these different options. You can have experiments that are hypothesis driven, and that hypothesis should always drive what it is you end up planning in your experiment. But at the same time, you can use a microscope to explore and see what is possible with your sample, see what is possible and what information might be available that you could turn into a hypothesis-driven experiment further down the line. So I want to reiterate that different goals of your experiment require different considerations, and because of that, they'll have different approaches to your experimental planning. So you've now identified that kind of what your objective of your experiment is. The next question is, what spatial information do you need to capture? Because that is the primary aspect of microscopy, is giving you spatial information. You need to understand how are you going to detect the specific thing that you're interested in your sample, and I'll get into a little bit more information about this now. And there's sometimes other information that you might need. Sometimes you might be interested in a protein, but it'll be very important for you to know where the nucleus of a cell is so that you can identify cell density. And that's not necessarily the most primary of the highest importance in your kind of objective, but it is something that you need to know from your sample. And then you should have an idea of, or try and begin conceptualizing what it is that you're trying to measure. So if we go back to the top of the list, what is the objective of your experiment? This goes back to my previous slide. What are you trying to do? Are you doing a pilot study? Are you doing a full-scale experiment? Are you trying to look at tissue specimens? Are you trying to look at cell culture? It all depends on what you're trying to achieve. The spatial information is important because that provides biological context. Sometimes that is related to you only interested in a specific aspect of biology that occurs in a very specific region of the sample, whether that's at a tissue level or whether that's at a cellular level. That is spatial information. Therefore, a microscopy experiment is important. Sometimes you only care about the mitochondria, which can only be nicely captured if the cell is whole and the full capacity of the sample is maintained. Therefore, microscopy is important. How are you going to detect whatever it is that you're interested in seeing? This is important because you need to be able to distinguish your feature of interest, whether it's a protein, whether it's a specific dye. There has to be a way of you capturing that information from the rest of the sample, because that's the fundamental principle of microscopy. So microscopes rely on separating these features, whether it's pretty straightforward in the sense that you want to identify if there is a cell or no cell, or whether you want to see whether there's expression of a gene or no expression of a gene, whether you're interested in something that's in the nucleus or cytoplasm. There's always a manner in which you have to differentiate what is in your sample. And this can be achieved in many different ways. Many different microscopes exist because of this exact need. And that includes H&E stains for tissues. You can use DIC microscopy to see the outline of cells so that you can count cells without needing dyes. Or you can use fluorescent antibodies or fluorescent proteins if you're looking at very specific components. So it all depends what it is you're trying to achieve. The other information that you need, as I mentioned, is your sample context. If you're interested in a particular biology, you might also need to know where the edge of the cell is. So you need a way of identifying the edge of the cell. So that's a consideration that you need to take note of. If you're doing tissue biology, it's very helpful to know where the cell nucleus is so that you can help count cells. That's spatial context that you need. And then what are you going to measure? Now you need to consider this with the caveat that microscopes really only provide two pieces of information. That is space or position or location, and then quantity. By the intensity of the signal, you can infer how much of your target is present, whether it's an antibody or whether it's a nuclear stain or whatever it might be. The real magic comes in how we combine these two measurements of space and quantity into interesting things such as density or shape or expression. That's where a lot of analysis pipelines come in. But keep in mind what you're trying to measure and how do you derive that measurement. So this is still part of the planning and initial conceptualization of your experiment. You should be aware of all of the components of microscopy. And there's generally three main categories in a microscopy experiment. The first being your sample prep, the second being your image acquisition or your imaging microscopy parameters, and then the last component is your data analysis. So the sample prep consists of everything related to the sample, where it came from, how it grows, how you have to prepare it, how you have to label it. The image acquisition relates to how many samples do you need to go through on the microscope to get the data you need? How many regions of interest do you need? Do you have to worry about how fast you're imaging if you're doing live cell microscopy? Are you interested in a variety or multi-parametric data where you need different channels? That all relates to your image acquisition. And then your data analysis kind of speaks to your final outputs and how you get there after your acquisition. So it includes just the images that you end up looking at, but it also includes those measurements I spoke of, a combination of location and intensity. The difficult part with all of this is that these three things cannot be considered in isolation. They have to be considered all together. You will find instances where your data analysis needs dictate what has to happen with your sample prep. In the same way, some of your sample prep restrictions will dictate what is possible with your data analysis. So you kind of have to maintain this holistic view of what you're trying to achieve with your microscopy experiment. Then the first bit of information that I'll bring to everyone's attention is that literature is your best friend. It's the first thing that you should do when you're considering planning an experiment, especially if it's the first time you're doing a new type of experiment. So you can gain from literature what other people have done. Have they done it on similar samples or relatively different samples to you? Why have they done it in a particular fashion? What was their goal? Are you trying to directly replicate what they've done? Or are you just trying to understand certain components of your biology? What type of microscope did they use? What did they do for their sample prep? And what ultimate information were they able to achieve using this microscopy experiment? And this applies to if you're doing very simple experiments or very technical experiments. The same kind of logic applies. See what else or what other people have done, and then try and replicate that and apply that to your experiment and your setting. So from the literature, you'll also be able to kind of get an appreciation for what to expect with your experiment. And that applies to the three components I mentioned before. It's your sample preparation, it's your image acquisition, it's your data analysis. These are all the three main themes that you'll always see in most microscopy experiments. And then you can decide, are you going to modify an existing protocol? Are you going to directly implement an assay to count cells that are stuck in metaphase of the cell cycle? Some of these things can be replicated and repeated, but some of them you have to kind of explore and work it out as you move along. So ultimately, let the literature guide you. And the literature, there's a lot that you can find. Not all papers will have fully comprehensive methods, but the methods are there and must be used when they're available. You can get information on what type of microscope they used to do this experiment, what they had to do for their sample prep, and then that can help you to understand what you need to do in your experiment. I'll also take some time to kind of differentiate two different aspects of bioimaging, because we often refer to it as microscopy, but it is bioimaging. You have one aspect where you're focusing on microscopy and you have another focus on biology. So when you're focusing on the microscopy, this is when you're still trying to do a technical microscopy experiment. You want to identify, is this experiment appropriate? Is it going to be to occur the way I want it to occur? Am I able to carry it out successfully? This is where your focus is on the microscopy, and the biology should stay relatively defined. And by saying that, that means that you get to play around with the sample preparation, you get to try different microscope settings, you get to try different analysis approaches and things like that. All of this focuses on the microscopy or the technical aspects of the microscopy. And your ultimate outcome is that you can understand how different microscopy parameters can provide different information and how you are able to interpret that information. You then contrast that when biology is the focus. This is when you want to keep your microscopy relatively defined, and it allows you to play in the biological space in a way. It allows you to change the biological variables, and by this I mean to use experimental groups to try different treatment conditions and compounds, to use different mutants or different treatment conditions, all sorts of different things. And this is where your focus is on the biology. And because the focus is on the biology, it allows you to link any measurements you detect are a result of that biology, and they're not a result of you changing the microscopy technical aspects. In combination, the reality is that we kind of have to straddle between these two different scenarios, where a lot of bioimaging happens somewhere in the middle, where you do have to tweak sample preparation and biology at the same time. Sometimes your sample preparation will change how your specimen is able to be stained, and sometimes we have to move between these two components. But if you want to build up, you want to ultimately build yourself into a situation where you as a researcher are confident in the ecosystem that you have to perform a microscopy experiment. And that can start by just simply defining your final goal. What are you trying to achieve with your experiment? And then through literature searching and through trial and error, you can identify what are your intermediate goals. And again, you refer to the three components of the microscope experiment: your sample prep, your microscopy or your acquisition settings, and your data analysis. You can let those intermediate goals direct pilot experiments where you can try things out, reiterate, change a setting, change a sample preparation method, and see how you can slowly optimize a final experiment where you would be able to perform a full-scale experiment. Some experiments you can get to this relatively quickly, and it doesn't require a lot of iteration. Some of them you don't know. You have to validate things. You have to understand what it is that's going on. How is it different? How whatever you're doing is different from the literature. But ultimately, I want you to understand that this is iterative. It is cyclical. As soon as you get to a point where you think you're finished, you can reassess your experiment and see, okay, what if I change this? Would I be able to get different information? So you can slowly build an ecosystem by understanding your pipeline and developing it over time. So when you move to actually planning your protocol, start by where are you getting your protocol from? Are you getting it from a collaborator? Are you getting it from the literature? And be ultimately guided by those initial points that are highlighted. What type of experiment are you doing? Is it a hypothesis-driven experiment? Are you just trying to see what your sample looks like or how it responds? Let that guide your protocol. But if you're doing an experiment for the first time, you are probably going to need to validate some things. You need to be able to understand, is your sample and is your microscopy experiment occurring the way you're expecting it to occur? Sometimes your antibodies don't work as expected. Sometimes the sample preparation doesn't work. Sometimes cell culture doesn't do what you think it does. And you need to go through these processes, or at least these steps, to identify what works and what doesn't work. And in essence, each of these validation steps is a mini experiment in itself. So you should kind of approach it the same way you do an overall project. So sometimes things don't work for the first time, and that's okay. You can adapt your protocol. you can troubleshoot, and you can optimize. These are all very important parts. Obviously, routine microscopy experiments have less troubleshooting, but particularly when you're doing something new or something for the first time, you might have to spend some time troubleshooting and optimizing. And that can be any part of your pipeline. It can be your antibody labeling, it can be your sample retrieval method. There's many aspects that this applies to. And then once you're able to gain biological insight, can you see the thing that you want to see? Are you able to measure it? Are you able to see a difference between an experiment and a control, for example? You can then scale up as necessary and then move into a full-scale experiment and get lots of data. You've got to consider your sample number. You've got to consider many experimental variables. And when you're doing any microscopy experiment, controls are very important. And you need them. You have to plan your experiment to include controls, because without controls, you don't really have an experiment. So there are generally two different types of controls, one of them being validation or technical controls. And these are mostly useful when you're validating something for the first time. And they are invaluable when things occasionally go wrong and you need to troubleshoot. You need to refer to your controls to understand where something is going wrong or where something is going right. And some of these questions include, is the labeling working? Did the sample preparation damage the sample in a way that isn't conducive to your experiment? And then there's also the more subtle and nuanced need for controls, your technical controls, in the sense that, are you capturing what really exists? Because the reality is that you have autofluorescence, you have background signal, you have crosstalk between fluorescence channels if you're doing fluorescence microscopy. All of these things can compound the validity of your experiment, and you need to understand them. And in order to understand them, you should do controls. And no stain control is very important. It helps you establish whether your protocol has worked or not. Single stain controls are also really important because they allow you to look at your feature of interest in isolation, and does it look the way it should look? Then on the other hand, you've got experimental controls, which you always need to include if you're doing a biological experiment. This allows you to establish baselines or minimums that can help you in identifying the correct parameters or microscope settings that you need. You also use them for understanding what might be the most signal that you need to capture in order to normalize your results appropriately. And ultimately, the controls are what provide comparisons in your biological experiment. And biology is a comparative science. So you have to be able to compare your experiment to a control, or you can't infer anything about what has happened. And these are your more straightforward or probably more familiar controls. These are your vehicle controls, your positive controls, off-target controls. There are many different controls for many different purposes, but you should include them in your experiment. I will quickly highlight that some of your validation controls, including, or in particular, your no stain control and your single stain controls, you might need them as part of your experimental pipeline. These can be for background calculations, for channel unmixing, all sorts of downstream applications. So don't overlook them. They can be very, very important. So I've just put up this text here because I want to reinforce them, that microscopy is complicated, and every single part of your experiment exists for a reason. And if you change any part of your experimental pipeline, you're changing the results at the end of it. And you need to understand how you're changing that, and that helps you understand how you should interpret that data. So controls are the tools that we need to understand and interpret our experimental results. So the next question is, do you have enough samples? So remember, each control will need its own sample. And there's different implications to this, which I'll cover now. And I also want to highlight that one specimen can sometimes yield more than one sample. So if you have one brain hippocampus, for example, you can section that into 20 different sections, which means you have 20 different samples to perform controls on. You also need to consider how many replicates you need. You need to establish your technical versus your biological replicates. You need to kind of understand your underlying biology and acknowledge how many samples you need to maintain statistical power and to ensure your statistical significance. But ultimately, this is balanced with the sample availability. I know that in animal studies, animals are not a resource that we can squander. So you have to maintain a low number of samples. Whereas if you're dealing with bacteria that you can grow in a flask, you can have many, many different kinds of samples. So it's always context dependent in this case. But ultimately, planning and preparation is a crucial aspect, and it's what is going to ensure that you can complete an experiment successfully. And I'll just go through that a little bit quicker. I know my time is running out, so I apologize. I'm going to skip this slide for now. And I want to highlight that sometimes we think of a microscopy experiment in the traditional way that it is taught in undergrad, where you have a hypothesis, you set up your experiment, you do your sample preparation, you do your microscopy, you analyze your data, and then you get results. But that sometimes, and more often than not, with microscopy in particular, is not the best way of approaching how to design an experiment. Sometimes you need to actually think about it in the reverse direction. So the easiest way to do that is, what is your hypothesis? And then the very next thing that you should think about is, what informative result would be most indicative of my hypothesis? What would be most informative, rather, of my hypothesis? And then work backwards from there. If that is the information I need, what kind of data will provide me with that information? And take it one step backwards. If that is the data I need, what parameters do I need in order to get that data? And then you can apply that to not only microscope selection, but also sample preparation. If I'm restricted in my parameters, what do I have to do for my sample preparation in order to design an experiment? And ultimately, you end up in this cyclical pipeline where you need to reiterate and you need to reassess how your sample preparation ultimately is reliant on what you want from your data analysis. If anyone wants to dive into this paper a bit more, you can find it by going to the DOI or taking note of the DOI at the bottom right of the screen. But if it is a little bit too complicated, just know that it boils down to the three main components that I've already established. It's your sample prep, it's your image acquisition, and it's your data analysis. And they are intimately linked, and you can't think of them in isolation. So I will quickly wrap up with my take-home points and say that it is very easy to get lost in all of the variables and the controls and amazing things that a microscope can do. So stay focused. Make sure that every experiment you perform has a clear goal, and make sure you stick towards achieving that goal. And bear in mind that every aspect of your experiment is important. If it's not relevant to your goal, don't do it. If it is relevant, it will affect the way that you can interpret your data. So make sure you understand why you're doing it that way and that you're doing it correctly. And get help from the literature wherever you can. Ask colleagues, ask collaborators, ask the ABIC community if you can for help and guidance. We're all in this together. We all want to help. It's also an important point to note that experiments don't always go according to plan, but that doesn't mean that it's a failure. Failed experiments can actually tell you a lot of information, and it's up to you as a researcher to learn from that information and adapt it into the next experiment and slowly progress towards your final full-scale experiment. Remember that biology is a comparative science, which means you have to have controls. So you need to plan your experiment around having the correct controls. And work step by step towards developing your full microscopy pipeline. Break down big experiments into small objectives if you have to, and treat each small objective like a mini experiment. And then don't ever forget this interrelation between sample prep, image acquisition, and data analysis. So that's the end of my talk. I'm not sure if we're having questions now or at the end.
We have questions now.
Great. I'm happy to answer any questions that there are in the chat.
Thank you, Mike. We have a question from Chukunonso, who said, you mentioned that you need to know how many samples that we need to achieve statistical significance. I know that for lab rats, a minimum of 4 rats are adequate, but for historical samples, how many sections would one need to assess to achieve statistical significance?
It's a very good question. Unfortunately, it's not one that I can answer up front because, as I mentioned early on in my short webinar now, is that it's very dependent on the exact biology that you're looking at. Some biological phenomena are very subtle, and because of their nature of being very subtle, you have to measure a lot of samples in order to confidently discern the differences. Some biological features are more apparent, and you don't need that many histological samples. So it is a bit dependent on the biology, but let some of the literature guide you. If other papers have got very good information and you think that they have strong statistics, work from that. If they did 30 different histological samples, let that be a starting point. It's also about balancing your technical repeats from your biological repeats in order to ensure your confidence in your result. So I apologize, it's not a very satisfactory answer, but it is a very context-specific question. So if you have specific questions, you can feel free to email me with the email address down at the bottom of the screen. But for now, I hope that is an appropriate answer.
Thank you, Mike. We do have another comment in the chat box. Just a few considerations more than questions from my side. This is Emmanuel. As a bioimage analyst who has worked for 10 years in a bioimaging unit, I also want to suggest starting with pilot studies and not starting right away acquiring hundreds of images and then finding out that sample preparation or image acquisition was wrong. Secondly,
Yes.
along with the literature, I just want to emphasize if you're lucky enough to have an imaging unit, consult it before starting the experiment. It can help a lot.
Yes. So I agree 100% with both of those points Emmanuel has mentioned. Never ever jump into a full-scale experiment. You have to build yourself up slowly, because the worst thing that can happen is that you now have hundreds of gigabytes of images and they're useless because they didn't capture the correct information you needed. So I'm of the opinion that you should go slower and steady rather than just trying to get to the end as quickly as possible, because more times than not, that ends up putting yourself at a disservice, in all honesty. And then the second point, yes, you can ask the literature, but if you have a core facility available and if you're fortunate enough, ask them. They're sources of information and experience. Ask people that have prior expertise in microscopy, and they'll be able to either answer your question or point you in the right direction at the very least.
Thank you, Mike. If we don't have any more questions, in the interest of time, I'll ask Soné to please start as well. Soné?
All right, let's try. I just lost my screen. Can you hear me and can you see my full screen?
See a full screen and we can hear you. Thanks, Soné.
Thank you. Good afternoon, AB community. It's my privilege to talk to you today about different microscopy techniques and technologies that you can use to study biology. My aim is to use the next 20 minutes to give you the important considerations about your sample and your experiments that will enable you to choose the right microscope to ensure that you get the best possible results for your research. But before we discuss different technologies, let's first start looking back into the past and look at the system called a microcinematograph that was built by Carl Zeiss in the late 40s. Looking at this image of the system, we can see there are many familiar parts. Of course, there's a microscope, and around the microscope, there's an incubation chamber to keep the temperature constant. And attached to the microscope via an adapter is the cinematograph, which is the machine that will help us to take a movie of our living sample. Now, one of the first movies taken with such a system, here we can see the cell division process of cells from a grasshopper using phase contrast illumination. And this enabled us to see microtubules and chromosomes as the cells divide. Today, we still look at a lot of similar processes, like in this example, where you can see a cell going through mitosis and the chromosomes being separated by the mitotic spindle. However, what has dramatically changed is that now we can label specific macromolecules, which we are interested in, by using fluorescent proteins, which have been developed in the mid-90s. And this has changed the world of microscopy in the most impressive way. One of the most interesting features of fluorescent proteins is that they can be used to label any sample, from bacteria to animal cells, and as we can see here, even plant cells. In this example, the microtubules are labeled in the root cells of Arabidopsis thaliana, allowing us to visualize processes inside cells. Furthermore, even complete organisms can be imaged, with one or more specific labels that allow you to observe any biological process you have in mind. In this video, we look at the embryonic development of Drosophila. Through labeling the microtubules, we can observe the cells dividing, but we see that this is not happening uniformly throughout the embryo, but some areas have more division than others. So in this talk, I would like to first define some of the key parameters you can consider when you are trying to decide on your imaging. And then we will discuss different examples of samples and look at the best microscopy techniques to use. But first things first, when you are imaging, you want to consider the signal-to-noise ratio. Signal-to-noise ratio effectively is how much stronger your sample signal is compared to the background noise. As you can see here in this image, the signal-to-noise ratio is getting progressively higher, enabling us to see a clear picture of Charles Darwin. A second factor that is tightly associated with signal-to-noise ratio is gentleness. And that is because often when you try to increase signal-to-noise ratio in your image, you would often increase light intensity or laser power. However, that can lead to photobleaching as well as phototoxicity. Numerous publications have shown that excessive light exposure can interfere with cellular processes and, of course, bleach your sample. So collectively, the first factor you need to think about is sensitivity, which includes gentleness and signal-to-noise ratio. The second imaging parameter to note is how quick is the process that you are trying to capture. Here you can see an example of beating of cilia in ependymal cells lining brain ventricles that requires very fast imaging at 143 frames per second. However, if your process that you're looking at does not require fast imaging, you should always try to take just the acquisition speed you need in order to keep the light dosage to a minimum, because remember, too much light can compromise the system. The third imaging parameter is the depth of your structure. For instance, are you looking at the top layers of an organ, or like in this case, are you looking deep into the brain tissue trying to capture long axons? Tissues have inherent ability to scatter light, and the deeper you go, the greater the scattering. So you really need to consider this factor. For the sake of time, I will not be covering a lot of technologies that consider the depth of or very deep tissue samples. But if anybody has any questions, you're more than welcome to send me a message or ask at the end, and I'm happy to discuss some of the technologies there. The fourth imaging parameter is the resolution. Resolution is defined as the minimum distance between two structures that would allow us to see them as separate objects. This practically means that if the structure you're looking at, which in this case is a neuromuscular junction, if the structure is beyond the resolution limit of the system, then the neuromuscular junction will appear as a circular object like on the left. Whereas once you increase the resolution, you can observe the true donut shape of the structure. The first imaging parameter is about how many samples you need to work with, practically the throughput of your experiment. Because imaging a single well in a 384 well plate will take a completely different amount of time to imaging the whole 380 well plate. Remember, if you need lots of samples, then automation is a key consideration. The sixth and last parameter you need to consider is, of course, the size of your sample. So do you want to image cultured cells or whole zebrafish or even live mice? For the sake of simplicity in this talk, I will define samples as thin, medium, and large. And we can consider them equivalent of maybe single cells, an organoid, or a whole zebrafish. So if you place the imaging parameters we discussed on a grid, we can use this plot to create, let's say, the perfect system that excels in all of these parameters. We can now use this as a scoring system for different imaging techniques. And I hope that this will be helpful to find the right system for your experiment. And I appreciate, and you should appreciate that you don't need the perfect system, which, by the way, does not exist. You need to identify which system will be suited best to your experimental needs. So when you are planning your experiment, you can consider these parameters and select the ones that for your particular experiment are non-negotiables, and then on which you can be more flexible. You can then use this in a way to select the microscope type that will be most suitable. It's important here to note that there may be an overlap in some of the technologies that I will go over now. However, my aim is to highlight the unique strengths of each method. So let's start by considering a first sample example in which you want to say image a thin sample, for instance, some cells, and you want it to be very sensitive and quick. The best method for basic fluorescent cell imaging would be to use a wide field microscope. Wide field microscopy illuminates the whole field of view and collects the image with a camera. You can choose to take an image of single cells or a single plane, or do a Z stack to get three-dimensional information. In either case, the captured image contains light from the focus area, as well as a significant amount of background signal from out of focus light above and below the focal plane. In order to remove out of focus light from the area or plane of interest, you must do optical sectioning. That is to apply specific microscopy techniques to remove out of focus light to produce high contrast images of the plane of interest. Here we can see an example of living dividing cells imaged with very gentle illumination at a very high speed with a camera that allows you to capture the whole field of view. As you can see, the image is beautiful on the left, but there's a lot of out of focus light collected. And one way to improve the quality of this image is to use deconvolution. What is deconvolution? It's a way to improve your image by processing it with software after you've acquired it. And in fact, it's a technique that's been well established for a very long time. Deconvolution can help you to resolve your image better, and it works best when you have loads of light collected, as in this case of the wide field image. So, as you can see here, deconvolution can help you to resolve microtubule structures better. And in our Zeiss software, we offer a fully integrated deconvolution module with an easy workflow and high processing speed that allows you to correct even advanced features such as spherical aberrations. To sum up, wide field with deconvolution can be the best option to use when your sample is quite thin, such as single cell layers, and you are interested in speed and sensitivity in your imaging. For our next sample, let's consider something thicker, such as a whole brain or a whole zebrafish, with optical sectioning and the needs for great sensitivity, or possibly even looking at multiple fluorophores at a reasonably high speed. This parameter combination will strongly support the use of confocal microscopy, which is one of the most versatile imaging platforms available. Confocal microscopy allows you to optically section thick samples, separate multiple fluorophores, and do photomanipulation experiments. As you can see in this example of the central nervous system in the cockroach, laser scanning confocal microscopy can acquire large volumes of data of multiple labels with very high resolution. So when you want to image a sample labeled with multiple fluorophores, sometimes you will find that some of the labels have emission overlap. A laser scanning confocal microscope can quickly solve the issue with spectral imaging and unmixing. In this example, you can see that the sample has multiple labels and strong autofluorescence since it's a living plant. And by using linear imaging and unmixing, we can quickly and easily separate all the labels and remove the autofluorescence. And because all the labels are acquired simultaneously, you are as gentle as possible to your sample, avoiding any overexposure or damage. In addition to spectral imaging, confocal microscopy technology allows you to precisely manipulate the light in order to capture dynamic processes. Here on the left-hand side, you can see an example of Kahidi photoconversion, which gives you a valuable tool in tracking cells, as well as a FRAP experiment on the right-hand side, which allows you to capture dynamic processes in cells. It's exactly the precision of a confocal microscope that allows you to do quantitative measurements of protein dynamics and interactions through fluorescence correlation spectroscopy. In a nutshell, FCS is monitoring the movement of single fluorescent molecules through the excitation volume, and the measurements you acquire can then help you to determine the molecule's quantity and movement dynamics, as well as their association with other molecules. To sum up, laser scanning confocal microscopy can allow you supreme flexibility in your imaging and a fair balance between all imaging parameters. So going back to our imaging parameters, let's now look at an example where you want to be sensitive and have flexible sample sizes, but you're really interested in very high resolution. When you are imaging with a laser scanning microscope, your laser beam is a single point. But in order to achieve optical sectioning, you use a pinhole to discard out of focus light. One of the ways to increase the resolution of your imaging is to close down the pinhole. But as you can see, this results in a significant loss of light. Zeiss have therefore developed the Aries scan detector, which allows you to combine the best of both worlds, with each detector element acting as a small pinhole, but the whole detector collecting as much light as possible coming from your sample. This can practically translate to allow you to observe the true structure of, for example, mitochondrial membranes, giving you super resolution in combination with a time series. To summarize, the Aries scan detector is practically enhancing the capacity of the confocal by increasing its sensitivity and resolution. But if you look at this graph, this comes at the expense of imaging speed. So what happens if you now want to have that speed? This Aries scan detector comes with a special software functionality called multiplexing, which helps us to mitigate this problem by effectively allowing us to reconstitute pixels in time. And here you can see how this reconstitution by a factor of 2 allows us to capture cell division dynamics without having to zoom in, giving us enough speed to capture the process and giving us the larger field of view. By playing around with these type of parameters, we can also increase the speed more. So in this example, where you need very fast imaging, like microtubule buckling in cardiomyocytes, here we see on the top left an image acquired by a resonance scanner. Even though the resonance scanner has the high speed, unfortunately, it doesn't give us the resolution to see the structures. Whereas the combination of multiplexing allows the researchers to get super resolution over a larger field of the sample and enables viewing of multiple labels. So practically, if you have a special detector with multiplex mode, then you can balance the confocal system to have a great combination of sensitivity, resolution, speed, depth penetration, and even throughput. So far, all the examples and the non-negotiables have revolved around the quality of the image, focusing on parameters such as sensitivity, speed, and resolution. But let's quickly change gears completely to show some examples of your work where the primary focus is throughput. And you might be a little bit more flexible on the image quality parameters of a thin sample. As I mentioned in the beginning of my talk, when throughput is your focus, automation is key. That is why a system like the Cell Discoverer, which is an automated platform with integrated environmental control and its unique optomechanical concept, is by far the recommended system for throughput applications, because more samples imaged means high reproducibility of results, more powerful statistics, and greater impact of work. With the Cell Discoverer, you can fully automate the wide field imaging of cells labeled with multiple fluorophores, with the system's special objectives ability to image an entire well of a 384 well plate in one frame in high quality that is comprehensive enough for further segmentation of the cells, and even factoring in how quickly you can now scan a full 384 well plate. If you want to look at medium-sized samples, such as example of live cricket embryos development, where the nucleus is labeled by GFP, you can image multiple crickets in a nearly synchronized way. And looking at the acquired images closely, you can even observe cellular events such as cell division and track individual cells while being super gentle to your sample. For applications that would require exceptionally long imaging, the Cell Discoverer is ideal since it completely controls the environment of your sample. Here you can see a single cricket embryo developing over time without bleaching and without light exposure interfering with the embryo development, allowing you to visualize developmental events such as dorsal closure as well as limb and body growth. To sum up, the Cell Discoverer is a quite unique system that can bridge the gap between wide field and confocal microscopy and can be very versatile depending on experimental needs. This type of system can also be fitted with the Aries scan detector to give you super resolution results. Sorry. So let's now consider some more special applications where, for example, you need super resolution but cannot compromise on gentleness and speed. Then the ELIRA platform will be the go-to system. We offer two types of ELIRA, a super resolution structured illumination system, or SR-SIM, as well as a single molecule localization microscopy system. Here, the SIM can give you quite impressive super resolution data since it's a camera-based system. So it gives you very high speed and very high sensitivity. This practically translates into a system that allows you to acquire really impressive super resolution information of multiple colors simultaneously. Like in this example, where we see beautifully resolved mitochondrial membranes and the microtubule growth as the cytoskeleton reorganizes. In summary, if you want to focus on gentle super resolution at high speeds, and you do not need to image at great depth, then the ELIRA 7 system is your go-to microscope. So the take-home message is that light microscopes are very diverse, and each system has its unique strengths and limitations. But the key to success for you is to consider your imaging parameters and to know the data that you want to get out of the system. This will enable you to decide which system can fulfill your needs better. If you would like more information on any specific systems, you're welcome to reach out to the ABIC team or to myself. Thank you very much for your attention.
Thank you so much, Soné. It was a wonderful presentation. We do have some questions here. We have a question from Emmanuel. He's asking, considering the difficulty of having access to microscopes in Africa, especially in certain regions of Africa, does Zeiss have any program or policy to improve the accessibility of microscopes in Africa? For example, on the contrary, in many institutes, including where I work, there are a lot of microscopes that are changed every now and then, but are still good and could be made available to other resource-poor settings. Maybe I'm too much naive and don't see the logistics or regulatory issues.
Maybe this is not a question. Maybe I can actually ask Michael Karen to answer that question. But yes, I know that there are programs. Maybe I can just do a little bit of comment. I've tried to cover the microscopy techniques and technologies which are available on the African continent in various imaging centers like BioRTC, like WACBIP, like the African Microscopy Center in Cape Town, and also even in places like ILRI in Kenya. But Mike, I don't know if you want to maybe comment on.
Yeah. Sorry, I was actually typing a response, so I'll just read what I've written here for now. So firstly, Emmanuel, another great question. Thank you. So at least on our side with AMI, we are looking at exploring options of distributing donated microscopes and finding a way of getting our hands on them and getting them to where they can make the most impact. We are finding that it is more complicated than we initially thought, as there are a lot of financial implications and things like that. But we are trying to find a best approach to tackle this, and we're looking for partners and trying to see if we can tackle this as a collective group. If you're interested, we can reach out, or you can reach out to us. We can set up a meeting to discuss. We currently have AMI's program for equipment exchange and reutilization, which should be announced in the coming month, as everything is intended, where we are trying to get donated microscopes into various, to whoever can utilize it best within Africa. But right now we don't have a mechanism for identifying equipment at institutions that are willing to be donated. We're still looking at finding the best way of tackling that. I don't know if you want to add to that, Soné.
Yeah, no, I think you've summarized it well. So, Emmanuel, just to comment, yes, Zeiss is active participant to this program, and we have some systems that will be going into Africa, but it will be facilitated through the AMI peer initiative.
Thank you, Soné. We have another question here from Sizwe. How do you see microscopy integrating with other technologies like genomics, proteomics, to provide a more comprehensive understanding of biological systems?
Ooh, I think, Mike, you've covered this in your, so you can add, but I see microscopy as sort of the center part that brings all of this into perspective. It is a complementary technique that enables you to put all the genomics and the proteomics stuff into perspective. It enables you to see what you actually sort of think is happening. And it can really put the molecular side of stuff and give you a little bit more information once you actually see this interact under a microscope.
Mike, do you have anything to add, or are you happy with that?
No, that's a great answer. I guess I'll just add and say that there's always an appropriate tool for whatever you're trying to do. And sometimes microscopy is the best thing, and sometimes microscopy is not the best thing. And sometimes you need to do both microscopy and something else to really understand what's going on. So I'm a firm believer of integrating microscopy wherever possible to supplement information and provide additional insight. But I'm also a big fan of having completely microscopy-based experiments where you can infer a lot of the things that flow cytometry might infer, or yeast to hybrid molecular studies and things like that. So it all comes down to, I feel like I'm giving a very bad answer, but it's the truth. It all depends on what you're trying to achieve, but also more importantly, what you have accessible and the expertise to support that.
Beautiful answers, you both. I see there's a comment. Hi, Claire. Yes, the LSM is currently the basic size confocal. So yes, I think that's all the questions, unless anybody else has any more questions. It is 3 o'clock on the hour. I just want to thank everyone for attending, and thank you, Soné and Mike, for your presentation. They were extremely informative. The recording will be available on the ABIC YouTube page, and we do have other episodes up and coming, so do follow our social media platforms and keep an eye out on the newsletter. Sign up for the newsletter on the website if you're not already signed up, so you can keep getting updated on upcoming initiatives. Thank you all for your time and attention today, and enjoy the rest of your day.